Hello, and welcome everyone to this podcast. In this podcast, we're going to begin our discussion of replication. That is how we take one set of DNA and faithfully copy it to produce a second and identical copy of that DNA. Let's begin by defining a couple terms and also talking about the major experiment that was done to show that DNA is copied in a semi-conservative manner. We start off with our one copy of DNA here, and we know through the process of DNA replication that we produce two copies of DNA. We call this original piece of DNA that is going to enter replication, we call that the template DNA. We sometimes will refer to it as the parental DNA. As the DNA is copied, this original template DNA is used as a template to produce the second copy. And we call the new copy the daughter strand. As we look at these two pieces here, I didn't indicate which was the template and which was the daughter strand because at the time of these experiments, they didn't know. They didn't know if this whole strand here was the parental and this whole one over here was the daughter, or if it was split in between. One of these strands was the template, one was the daughter, one was the template over here, one was the daughter. They didn't know. They had three models though. So let's describe those three models. The first model was called semi-conservative. The second model was called conservative. And then the third model was called dispersive. In all these models, we start off with a piece of DNA. And we're going to end up with a new piece of DNA. In the semi-conservative model, what happens is these two strands that are wound together in the double helix are pulled apart. And I'm just going to draw them as a straight line, like so. And I'm going to put a T under here to indicate that these are the template strands, or the parental strands. They are the original ones that came from this double helix. Then through the process of replication, the template strand creates a new strand, the daughter strand. I'm just going to make it a different color here. Put a D there for daughter so that we can easily tell these apart. I didn't draw these as double helices, but you should recognize that indeed these are double helices. In this model, it's called semi-conservative because in each of the product, half of the original molecule is conserved. So it's half conserved, semi-conserved. Again, each new product here has half of the original strand and the other half is a new strand of DNA, the daughter strand. In the conservative model, and I'm just going to continue to draw these as straight lines just to be consistent with over here, the conservative model says that you take the whole original double helix and you use the double helix in some manner as a template to produce the new daughter strands. In this model, the original template DNA is fully conserved in the result. And then the new strand is completely new, two daughter strands. The dispersive is kind of interesting. And essentially what the dispersive says is that we're going to chop up the double helix and then use it as a model, as a template. And then once it's chopped up, it'll be reunited with new pieces of DNA. So in this model, what we're looking at really is a product that is both template and daughter strand for each of these 4 strands of DNA. They're a combination of them all. So now the question becomes, which of these models is correct? And how could we figure this out? In order to figure this out, we need a way, some kind of mechanism, to look at our strands that are completed after replication and be able to tell the difference between template DNA and daughter strand DNA. And that's what we're going to describe next. The experiments that were done to show which of these models was correct were done by 2 scientists, Meselson and Stahl. At this time, it was well known that DNA was composed of nitrogen-containing bases. The adenines, the cytosines, the guanines, and the thymines. They all had nitrogen in them. At this time, they also knew that nitrogen existed as 2 different isotopes. A heavier isotope called N15 and then a lighter isotope called N14. This was the lighter isotope. They knew if they grew an E. coli cell in the presence of the heavy isotope, and you let this grow for many generations, hundreds of generations, that nearly all of the DNA would be composed of this heavier isotope. And they knew if they isolated the DNA, they opened up the cells, that they would have a collection of DNA that was of the higher isotope, so a heavier DNA. Overall, this DNA would be heavy. As you can see, once again, E. coli is a workhorse in understanding how genetics works, particularly as we look at molecular genetics, as we've been looking at over the past couple podcasts. Anyhow, if we take this E. coli cell and we grow it in N14 media containing the lower nitrogen isotope, and then we isolate this DNA, and collect it, then we'll know that the DNA we have is lighter because it has the N14 as part of the nucleotides. But how do you measure this? What they would use is something called a cesium chloride gradient. And this allows molecules to settle at their density. So if we took 2 of these here, both of these are still cesium chloride gradients. Cesium chloride is just a salt. And if we applied our heavy DNA, this DNA 15, so we put N15, but it's the DNA made with the N15, and put it on this gradient, and then we took the lighter DNA, the N14 DNA, and applied it to the gradient, and put these tubes in an ultracentrifuge. And in this ultracentrifuge, let me go ahead and spell that here. And once in the ultracentrifuge, they would spin these tubes very fast. So 100,000 RPMs. And you would let this go overnight, 24 hours. During that time, while you're at home sleeping, this machine's doing all the work for you. During this time, the heavy DNA is migrating through the cesium chloride gradient. The lighter DNA is also migrating through this other tube. In the morning, you come back and look at your gradient. And what you discover is, within this gradient, the 2 pieces of DNA have migrated to different levels. The N15 DNA, the heavy DNA, has migrated closer to the bottom, whereas the N14 DNA, the lighter DNA, has not gone as far. It's less dense. Now what happens if we take a single tube, single gradient, and we put both N15 and N14 DNA in it? So let's erase some of this now. So again, we'll start off with our same cesium chloride gradient, but this time we're going to put in both the the heavy DNA, and then DNA isolated from the bottom one here, the light DNA. You put it in the ultracentrifuge. It spins 100,000 RPMs for 24 hours. You come in the next day, and what you see in this gradient is a separation of these. We have our heavy DNA at the bottom, near the bottom, and then we have our lighter DNA near the top. So this proved to be an effective way to look at a single culture and determine if the DNA in it was heavy or if it was light. So let's go over the Meselson and Stahl experiment. So Meselson and Stahl, what they did first was in a flask, they grew the bacteria in media that contained only N15. And they let this go for many generations. E. coli has a generation time of about 20 minutes, 17 to 20 minutes. As an interesting side note, sometimes we think that all bacteria grow really fast. It turns out some bacteria grow painstakingly slow. It can take days for them to divide. So it turned out E. coli again was a really good model for this because it grew pretty rapidly. And at this time we understood, the scientists of the day understood how to manipulate it and grow it in certain ways, and so it was a very useful organism, and still is. After several generations, what they did then, so after these many generations, in this flask here, the cells were at saturation. They'd used up most of the media there, and so the scientists were fairly convinced that all the DNA contained this heavy isotope, all N15. They then added to this an excessive amount of media containing the lighter isotope N14. And they let this grow for 1 generation. 17 to 20 minutes. And then they isolated the DNA and put it on the cesium chloride gradient. I ran out of room here, but then what they would do is, I'm just going to draw an up arrow here, they would let it go for a 2nd generation, then a 3rd, and a 4th, and so on. And each time they would isolate the DNA at the end of each of these generations, and then place it in a cesium chloride gradient. Okay, let's analyze their results. Over here I want to write down kind of a key. So here's a cesium chloride gradient. So you would expect N14 DNA to migrate here, and you would expect N15, the heavier one, to migrate here. So that's just our control, to give us our standards of where things should be migrating. At generation 0, I'm just going to put G0 for generation 0, which is here, before you put in the N14, all the DNA migrated right here, just as you would predicted where the N15 is. Then after 1 generation, all the DNA migrated here. So higher than the N15 line, but lower than the N14 line. I'm going to purposely draw these lines pretty thick here for now. So this is generation 1. So let's let it go for 1 more generation. G2. And what we see at G2 is still this middle band. I'm going to try to draw it a little less thick. And then we start to see a band forming up here where the N14 would be predicted to be, judging by our key here. And after the 3rd generation, this band here gets even lighter. This one up here gets a little darker, but still this middle one in between 15 and 14, and this lighter one at N14. And let's see, G4, I'm going to have to shift it down here a little because I ran out of room a bit. But you should get the idea. Again, we still see this middle one, but it is very, 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 very faint. And this one up here, where the N14 is labeled to be, is even thicker. So let's interpret these results as best as we can. Our time 0 is made up of all N15 DNA, so I'm going to just write this in green. So over here, let's go, N15 is green. Any new DNA, let's make that purple. So N14 will be purple. After 1 generation, we see all the DNA is somewhere in between. So we know all the DNA here is composed of both N15 and N14. Well, let's see if it supports any of our models. Let's look at conservative first. Conservative says that we will take our original DNA molecule and use that whole molecule as a template and produce another molecule separate from it. If this were true, we should have half of our molecules at N15, the heavier isotope, and half of our molecules at the lower isotope, N14. And that's not what we see here. We don't see a band here at N15 and a band here at N14. We see everything in the middle, telling us that the new DNA has to be some kind of combination of the two. So it can't be conservative. Next let's look at dispersive. Dispersive says that we're going to have each of our DNA molecules containing components, each of our strands, I should say, containing some new and some old DNA. You would make a prediction if the dispersive model is correct that each of these molecules should weigh somewhere in between N14 and N15 because it contains copies of both. So this result supports that it could be the dispersive model. Well, let's look at what it would look like if it were semi-conservative. If it was semi-conservative, remember, it rips these strands apart and uses each half as a template. So let's separate these out. So each of the new models would contain half of the old, one strand that's old, and one strand that is new. And so this was the semi-conservative model. And so if this were true, you would expect each of the new molecules to be half N14 and half N15. And so they should migrate in between 15 and 14, and that's what we see. So at the end of the first generation, we can be confident that it's either the dispersive model or the semi-conservative model. Now let's look at G2. And I'm going to erase this semi-conservative model here and ask, what would the G2 generation look like if DNA was replicated in a dispersive manner? Again, this model would suggest that we would take this here and chop it all up and create new DNA. Since this is what we're starting with, and the only nitrogen present is N14, the new molecules will still be a mix of N14 and N15, but it will have, over time, less and less N15 in the molecules, and more N14. So to show that, I'm just going to draw this molecule here with more N14, more purple in it. There will still be some N15 because it's the strands are still there that had it, and you still have your template strand portion here, but it's going to be less and less. Because of that, it will be lighter. So what we would expect to see in the result, if it was by dispersive, is that this band would get lighter. So we might see a band like right above it, but that's it. So I'll draw that down here. We would expect the band, where the original band was here, but the new band, after the second generation, would be right above it, and we wouldn't see this other band. We would just see this one that's a little bit lighter. And in each generation, what we would see, because in each generation the resulting DNA would have more and more and more of the new lighter isotope. So this band would steadily go up at the end of each generation. But is that what we see here? No, we don't. We continue to see this one in the middle, and now we see one fully at the N14. So it's not likely to be the dispersive model. So let's erase that. And let's go ahead and erase the word dispersive just so we don't get confused. And we'll erase that molecule as well. And let's redraw the molecule as it would look if it were semi-conservative. Okay, so at the end of the second generation, what's going to happen is we're going to take this molecule and we're going to rip it apart, or rather the cells will do that. We won't, but the cells will do it. And you'll end up with two strands as the template. One that is in 15, the green one, and one that is the new one, the N14. Now each of these now will be the new template strands. And when they're making new DNA, they're only going to be able to use the heavy isotope, N14. and as a result, the molecules will look like this. So you'll have one that is fully N14, which we see here, and then we'll have one that is still half of each, which is here. This makes sense for a semi-conservative model. What happens in the third generation? Well, once again, these two molecules here drawn will be ripped apart and each will serve as a new template. So you'll still have this green one, but now we have three purple ones as templates. So let's just draw it like so. The new DNA that is going to be placed here, the new daughter strands, still will have to use the N14 DNA, nucleotides, the lighter nucleotides, to make the new DNA. So this one here. And as you can see, what's happening is that the DNA molecules that are mainly N14 begin to predominate. And that's why this band got a little thicker. We will always maintain some hybrid form of N14 and N15 together, because every time it divides, this strand here is going to be pulled apart and each half will be used as a template. This green one will, in the next generation, will once again be a template. But over time, this hybrid, the N15, N14, will become a much more minor component of the overall DNA. And the new strands that are being templates are already N14, and when they make new DNA, it's also going to be N14. So this here, the N14 only DNA molecules, will become much more predominant. So by analyzing these results, I think we can be confident in saying that DNA replication, as shown by Meselson and Stahl, is done by a semi-conservative mechanism. Sort of as like we did with the experiments showing that DNA was the genetic materials, ask yourself what the results would have looked like through 4 generations if replication was by a conservative mechanism or if replication was done by a dispersive mechanism. So let's summarize real quickly what was in this podcast. You should know the difference between a template strand versus a daughter strand. You should understand 3 models of DNA replication. Semi-conservative, conservative, and dispersive. You should know that DNA replicates using a semi-conservative mechanism. And then you should understand the Meselson and Stahl experiments. How what they saw showed that DNA replicates in a semi-conservative mechanism. That's all I have for this podcast. In the next podcast, we'll start looking at the enzymes that are used to faithfully replicate DNA. If you have any questions, please let me know. See you in class.